Well, I think we should start by now. And it's a great pleasure for me to introduce today's guest speaker. Everybody knows Dr. Brian Kennedy from the Buck Institute. And as most of you know, Dr. Kennedy has done many breakthrough works in aging research. One of which is the determine the role of the mTOR on the aging and the age-related diseases, which he's going to talk about today. And Dr. Kennedy had a PhD degree from MIT, and followed by the postdoc training in Massachusetts General Hospital. And although he likes the Boston, he was tired of the brutal, the cold winter over there, and he thought the moving to the West Coast could change his life better. And he went to he had the faculty position, the University of Washington, Seattle, and he spent another decade over there. And as you know, he has done so many interesting work over there. But the one problem is that he started feeling tired of the gloomy weather over there. And he thought he should better move to the sunny California. And then he moved to the Buck Institute. And as I said, he has done so many the breakthrough work using the various the animal models, which is from the Yeast and the mammals. And I'm very excited to hear his work, especially mTOR and aging today. But for some reason, I thought he's from somewhere in the Pacific Northwest. And I learned yesterday he's from Louisville, Kentucky, where the greatest boxer in the history was from. And no wonder the Brian is one of the greatest, if not the greatest, the researchers in the scientists in the aging research.
You're setting me up to fail here.
Thank you, Brian.
Thanks. No, it's a great pleasure to be here. And I got my start in yeast genetics, and just recently I received an IRB approval to do a human study, and so I think it's very dangerous to let yeast biologists do human studies, but we'll see where that goes. The one thing I didn't ask about is a pointer. Is this a pointer? This is a pointer. Great. Can we? Maybe it won't be too bad. The other problem I had, and I realize this is my fault, is I've been giving talks in various universities the last week or so, and I woke up this morning and thought, my 8 introductory slides on aging is probably not so necessary here as it is in other places. And it's a little late to change it. So I'm just going to go through and make a few points about that and then get into the research. I think that, to me, when I talk about aging, I think the most impactful slide I have is this one, because it really differentiates between two things. One is life expectancy, and the other is healthy life expectancy, or health span. And this points to the real problem in healthcare right now, which is that, life expectancy is going up. It's gone up in U.S. 4 years in the last 20 years if you're a male, so you're getting a 20% return on investment, essentially. For every 5 years you live, you can expect to live 1 year longer. But healthy life expectancy is not going up at the same rate. And I think that, I would argue, and I think that a lot of you might agree with this, that this is partly due to the fact that we're really not dealing with prevention or dealing with the aging process at all in our treatment. We're waiting until people get really sick, and then we're doing a good job of keeping them sick longer, and that's extremely high cost, and that's one of the things that's driving the healthcare industry out of control. And so I think, when I think about aging research, I think the goal really is to focus on this number and try to make this number go up at the same rate as this number, because if that happens, that really changes the equation. It becomes an economic victory, reduces healthcare costs, and improves quality of life. One of the other things I've noticed from going around the world talking about aging is that there's really, we need to take into account the differences in different populations in terms of risk factors. And so I used this slide recently. These are countries I've been to recently, were Singapore and Chile. And we're looking at different risk factors for mortality in these countries. And you can see that different countries have different problems. So if we take Chile up here, which has, by the way, a longer life expectancy than the United States, which is another story that we can go into. U.S., by at least one ranking I looked at recently, is 50th in life expectancy now. Chile has a big problem with smoking still, and so chronic obstructive pulmonary disease is very high there in the aging population. U.S. still has the market corner on obesity out of these three, and that's causing a lot of metabolic disease and aging. And then Singapore is doing quite well, really, in all of these rankings, and that's one of the reasons they're the second longest lived country in the world. Now, they have a happiness score on this, and frankly, I have no idea how you measure happiness. And I have 2 points about that. One is the United States apparently ranks 13th in happiness. And if you look at what's been going on either before or after the election, I can't imagine that we really rank 13th in happiness right now. Also, I was giving this talk for this big opening of this aging center in Chile. And Chile ranks pretty high in happiness too, 23rd. And I mentioned this. And the president of Chile was there. And she looked over at my colleague and she said, "There's no way Chile can be that happy because everybody hates me." So I don't know about this number, but these other ones I think are still relevant to aging. And of course, this is the argument I give to a lot of politicians when I talk to them, is that when you look at disability-adjusted living years in the developed world, or even the whole world right now, there's really three kinds of boxes. There's a green box, which is injuries, harm, things like that, road injuries, etc., falls. There's a red box, which is infectious diseases, iron deficiency, AIDS, HIV. And there's a blue box, which has a whole lot of different names on it: stroke, heart attacks, cancer, COPD, Alzheimer's. I think in large part you can take all these names off and put aging on them, because aging is the biggest risk factor for the vast majority of the things in the blue boxes. And so you talk about governments, they're really focused on aging now because they have a large percentage of their population getting old. And you ask them what they're doing, and you hear things like this: increased assisted living, senior centers, better aging in place. That's the catchword of all the government officials right now. More hospitals, retraining, monitoring devices, rehabilitation centers. This is where most of the governments are putting their money right now. And my point is that why don't we take a step back and do this? Why don't we just try to focus on aging and figure out a way to keep people healthy longer? And I really believe that it's going to be easier to target aging than it is to treat a lot of these chronic diseases of aging, which are really complex in nature. And the way I think about it is maybe too naive, which is that I see aging, the healthy aging, as sort of maintaining homeostasis. So you have events happening as you get older. You're getting damage to various molecules. You're getting changes in certain pathways, but your body compensates very well and more or less maintains a homeostasis until a certain point when it's not possible to do that anymore, and you sort of spin out of equilibrium, and that's when you get chronic diseases. And depending on which way you spin out of equilibrium, then you get different diseases. To me, once you're in that state, in a disequilibrium, it's really hard to put things back in the box and get back to health. But while you're still doing in that homeostatic state, it's going to be a lot easier to maintain it longer. And that's really what I see aging as doing. I tell you what I'm going to do. I'm going to take this jacket off. Is that going to bother anybody? I brought it. I have one. No. I'm going to have to. Can you hear me without the microphone? Is that? All right. Oh yeah. Okay. So let me change that. But of course, it's not just about disease. It's about function too. And so I compiled this data recently. This is a lot of people have done this. This is women's record marathon by age. And to me, you show people this and they say, "Oh, look at this. This is aging." And first of all, I have 2 points. One is if you're over 90 and you can finish a marathon, that's pretty good. But the other is that, to me, this is what aging is here. It's happening all the way through the process. And so you're getting a slow functional decline in a lot of different things during the aging process. This is a functional measure. You can take a cognitive measure. I just picked chess rankings because my older son plays chess. This is the average ranking of the top 10 U.S. players by age group. Here's just overall. This is not that high compared to a lot of other countries, although the U.S. is getting much better at chess because we're actually importing people from other countries now to call them U.S., and our chess rankings are going up. But if you look at people over the age of 65, they're somewhere between 13 and 14-year-olds on this chart. So, I think you can measure this with anything you look at, but to me it's not just about disease, it's about functional decline. And it's really about this sort of oversimplistic view here, which is that instead of declaring war on diseases all the time, obviously we need to work on these diseases. I have no problem with that. If I get sick, I'm going to go seek help. But we really should be focusing more on the thing that's in common between all these diseases. And I think the reason I say this is naive is this kind of suggests that aging is one thing, and I certainly don't believe that. I think this slide this review by David Gems is actually a good way of looking at it. Most of the world is still taking this approach where you have all these different terrible diseases and they all have their own causes, and we need to focus on them as if they're different things. What I showed is kind of this, which is aging is one thing that's causing all these diseases. But I think this mixed model is a really kind of effective way of thinking about it, which is there are several different things that are causing aging. If you look at the reviews, one person says 5, one person says 10, several different things that are causing aging, and they probably contribute differentially to diseases. So you might have this mechanism 2 here, which is contributing to 5 chronic diseases of aging, 3 of which can be life-limiting, so you get an effect on lifespan in the population of animals if you just try to target this mechanism. You might have another mechanism that only contributes to 2 diseases, and no, those are not life-limiting, etc. So this is kind of how I think about aging. And, we got together in this geroscience initiative about 2 years ago and tried to write a review about what causes aging, and we had these 7 things to focus on. And I won't go through them all. I think that the thing that was striking in the discussions, and I tried to put the review together, was how connected they all were. So to me, this is how you could have multiple things causing aging, but it really seems interconnected because you can start with dysfunction in any of these places, like metabolism, and get rapidly to all the others very quickly. So I kind of see aging as a network, and once you perturb the network enough, it starts triggering problems with a whole bunch of different pathways. I also think this is probably the sad part of where we are in the aging research field, is that we still are not at the level where we have a really good understanding of what are the events, the actual molecular events that are driving aging, and how do they fit together. I still think that's a problem, and maybe that reflects the complex nature of the aging process. Having said that, I think the upside is we've been very good, and the ITP program has been a big part of this, at identifying interventions that affect aging. So you have a lot of behavioral interventions like calorie restriction, exercise, fasting. I put alcohol on here just to be provocative. And you have a lot of small molecules that are now increasingly affecting aging, mostly in animal models. And so this is the exciting part. And I really believe we're at a point where we can intervene in human aging. We have so many different possibilities now, many of which are feasible in humans, that we should be testing them. And so one of the things that I want to do is try to help getting things tested in humans so that we can begin to understand whether all this information we're learning is valuable or not for human health. So I'm going to talk about really a little bit about some of the yeast work we've been doing, but then focus mainly on the mTOR pathway in mammals. So in yeast, I've been working on yeast aging for a long time now. And in about 2005 with Matt Kaberlein, we decided to try to figure out the lifespan of every deletion in yeast in replicative aging. So this is the system where you have the mother cell that divides and you remove the daughter cell and count how many times the mother cell divides. And we wanted to look for deletions that were long-lived. Now, we knew there were a few at that point in time, but there weren't that many. So the question was, if we looked at the whole genome, how many would there be? So there's a couple points to make. One is, if there's 5,000 non-essential genes, we found 238 knockouts that were long-lived. That's a lot. That's almost 5% of the knockouts. And actually, it's probably closer to 300 because by the way we did the statistical analysis, we knew we missed about 60 or 70 of them. Not that hard to extend lifespan. Now, that's yeast, I agree, but it's not that hard. Now, worm RNAi screen suggests pretty much the same thing. It's not, there's lots of RNAis to genes that extend lifespan, somewhere near the same percentage. And so one of the things we were able to show from this study, by the way, I cut this short. We had, I think, 88 authors on this. So we went through undergraduates galore doing dissections to remove daughter cells from mother cells. And so there were a lot of optometrists in Seattle and in Marin who loved the fact that we were ruining the eyes of the young people in the universities. One thing we learned from this is that if you take a knockout in yeast that's long-lived and you find the worm ortholog, it's more likely by about 5 times to regulate aging in worms than a random gene in worms. So that says we took 2 species that are very different from each other, and yet they're conserved pathways that modulate aging. So I think that's important. I think that's really the first quantitative evidence for that. Second of all, of course, we hit pathways that impact aging, like chromatin modifying complex. Here's metabolism, ER stress, and protein modification in the ER, translation, activation of GCN4, and we published on most of these things individually. What we're trying to do now, though, is sort of 2 things I'll tell you about briefly. One is go from genes to drugs and begin to understand drugs, and I'll tell you why we're doing that. And the other is to try to think of this from a systems biology perspective. We want to use yeast to understand how aging works in a whole organism, not one gene at a time. How does everything fit together into some holistic understanding of what aging is? And I don't know if we'll get there or not, but I'll tell you a little bit of the work on that. At first I was really hesitant to go after drugs that affect aging in yeast because I thought the chances that the drug would be conserved effects in mammals would be less than the genes. And it's also hard to screen. But I worked with Michael Palominas at Texas A&M, and we developed this cell cycle strategy to find drugs that we think, based on their cell cycle properties, might affect replicative aging. I can tell you about this if you're interested. It's interesting, though, that in the screen for drugs, the first 2 we hit were ibuprofen and gemfibrozil. Ibuprofen you've heard of course. It's an NSAID. It's a bit surprising that it affects aging in yeast because it doesn't have cyclooxygenase. And so the fact that ibuprofen had an effect suggested there was a different mechanism. And so we started looking into this. And it turns out that what ibuprofen is doing is it's affecting degradation of the tryptophan transporter. You can see that here. So if you add ibuprofen, you can stimulate degradation of the transporter, and that reduces tryptophan uptake. And in fact, that causes an amino acid response so that a large number of amino acids are underrepresented in yeast with ibuprofen, even on replete media where they have access to amino acids in the media. One of the reasons you have such a big effect on tryptophan is that it's one of the most expensive molecules to make. So if you have to make it as opposed to transporting it, it costs a lot of energy. And so if we took out the transport pathway and forced the cell to make its own tryptophan, it makes a lot less of it than you would have if you can get it from the cell. And we could show that that was important for the lifespan regulation. And interestingly, that's one of the things mTOR regulates too, is turnover of amino acid transporters. And then we looked at gemfibrozil, and it turns out that it's impairing growth on dipeptides. So we looked at different carbon sources where these drugs conferred growth defects. And what was unique about gemfibrozil is it doesn't grow well when you supply it with dipeptides, which are really abundant in the yeast media that we put them in. And it turns out that we can see that it's affecting the activity of dipeptide transporters. This is a long slide. Basically, when you knock out these dipeptide transporters, you no longer get lifespan extension by gemfibrozil. So these are 2 different drugs that are acting through different mechanisms in yeast to affect aging. Then there are known mechanisms in humans. And we're trying to understand now whether these pathways are conserved in mammals or not with these drugs. And what it's pointing to, though, I think, is the importance of limiting amino acid uptake as a regulation of aging in yeast. And so we're very interested in that. I would also say we're not going to show this data, but if you look at studies from that we're collaborating with Hao Li at UCSF, studying mechanisms by which calorie restriction extends lifespan, one of the big impacts we see when we look at an unbiased way of calorie restriction in yeast is it reduces the expression of methionine biosynthesis genes. And so you restrict for glucose, but you end up getting a methionine restriction in yeast. And so we think these pathways are very intimately linked. If you think of it from the systems perspective, we're doing kinds of studies like this. So here we've taken a large number of interventions that are long-lived. These are all knockouts except for overexpression of SIR2, the deacetylase, or calorie restriction. They all extend lifespan in yeast. And we want to know how many different pathways do these knockouts represent. And so what we're doing is combining them with mutants that block various pathways like GCN4 or autophagy. Here's a transcription factor for mitochondrial genes. This activates the proteasome, etc., making all these double mutants and then doing the lifespan of the double mutants. And then trying to cluster them to see how many different pathways can affect aging. And one of the interesting things is we got kind of a surprising result. We think if in this smaller grouping, we have three kinds of clusters here. This group here is kind of what we expected. So if you pick a long-lived mutant, like the RPL mutant here, it's pretty long-lived in most of these backgrounds, but it doesn't work in this MSN2/4 background. So that's unique. And it's blocked to a large extent in the GCN4 background. So these kinds of mutants have like one or two pathways that they require to extend lifespan. And that's kind of how we think about aging. We mutate a gene, and it turns up some stress response pathway, and that's good for lifespan. We knock out DAF2 in worms, it turns up DAF16 activity, and that regulates aging. And so we have some like that. But we have two other clusters. We have this group here. These are all long-lived interventions, but they seem to be blocked by knocking out a whole set of different pathways. So it's not that they require one stress response pathway, they seem to require all of them. And so we don't really know how to think about these. They seem to be changing the stress response state of the cell and generally in a small way, but inducing a bunch of pathways that interact with each other in some way, and they're all required for the lifespan extension. No idea what's going on here. Then we have two or three mutants here. We actually have more of these now. They pretty much extend lifespan in almost everything. And we think these are just refractory. So maybe they're reducing the actual damage that causes aging so that you don't need the stress response pathways anymore. So this is a naive way of thinking about things, but we were hoping to get things that clustered around autophagy or GCN4, but we get a few genes that do that, but a lot of them are behaving very differently. And so I think the way we think about how to modulate aging may be too simplistic. And so hopefully, our grid looks like this now, and we've got more clusters, and we're beginning to really do some statistical analysis and bioinformatics to try to put this together in some way that's meaningful. But it's a lot of data we've generated here, and hopefully at the end of the day we'll be able to look at it and really learn something about how the whole organism is regulating aging, not just one gene at a time. Now the main reason I got interested in the mTOR pathway is that when we looked at those things that were conserved between yeast and worms, the thing that really jumped out at us was mTOR signaling. So here's the knockout of S6 kinase in yeast, and it's long-lived. This is an RNAi for S6 kinase in worms by Milena Hansen and Cynthia Kenyon, and that's long-lived. And you can see that there's similar findings all the way up and down this pathway, and given that this is one of the pathways downstream of calorie restriction, we thought, well, there's a good chance this is going to work in mammals. And of course, Dave got very interested in the mTOR pathway for aging as well, and many others. So we've been really focusing on this in the lab. I don't need to tell you much about rapamycin here, probably. I'll just hit the highlights for anybody who needs it. So it's very responsive to nutrients, growth factors through insulin IGF signaling. By the way, interrupt me if you feel like it. And amino acids through the RAG complex. It's also responsive to stress, energy deficit, hypoxia, and other kinds of stresses. So this is very much sensing the environment, this TORC1 complex here. And it's telling the cell what to do by regulating growth and proliferation and also stress response pathways. And the beauty of this is that you have this drug, rapamycin, that is a very specific inhibitor of the mTOR kinase because it's not an active site inhibitor. It forms a trimolecular complex with FKBP12, which disrupts TORC1 activity And it's very specific as kinase inhibitors go. There's also a TORC2 complex, which regulates AKT inside a skeleton, is less understood and less linked to aging. But one important note is that if you give cells or animals rapamycin long enough, you can get TORC2 inhibition as well, and we've worked a lot on trying to understand what the difference is between inhibition of TORC1 and TORC2. And of course, you know this already, there's toxic side effects of rapamycin, and David Sabatini published about a few years ago that a lot of the metabolic side effects from rapamycin come from inhibition of TORC2 and not TORC1. So either TORC2 inhibition in the liver or the fat leads to altered glucose and hepatic metabolism. And that led us to think, I wonder if we could find drugs that only inhibit TORC1. So the cancer field has kind of gone in the other direction, look for active site inhibitors that inhibit both TORC1 and TORC2, but those have toxicities too. And for aging, we were thinking, let's only get TORC1. Is there any way to do that? So we took a very naive approach, worked with a company, got access to a bunch of derivatives of rapamycin that had 2 to 3 changes on the molecule, and just screened them in cell culture one at a time to look for things that inhibit TORC1 and don't inhibit TORC2. David Sabatini didn't think this would be possible to find these. He had a very, I think, plausible conceptual reason why you wouldn't be able to modify rapamycin to get this effect, but empirically you can. Because we found a bunch of them that behave like this. This is part of a company now. And this is the Western blots looking at S6 phosphorylation versus AKT. We've looked at multiple substrates of TORC1 and TORC2, and it's more or less the same. Here's the graph. TORC1 inhibition for one of our compounds is almost the same IC50 in cell culture as rapamycin. But for TORC2, it takes a 100-fold higher dose of the compound, and you never get to more than 50% inhibition. And actually, when you put these in the animal, we rarely see any TORC2 inhibition. This level of activity is so low that we can't get in vivo concentrations high enough to really see TORC2 inhibition. So it's very TORC1 specific. And we're very interested in pursuing these molecules now, and we have 2 companies, one looking at autoimmune disease and one looking at neurodegenerative disease. One of the problems with targeting TOR is that there's almost too many things to choose from, and it's really hard to pick which indications are ideal for your particular compounds, and that's a challenge. I'll just show a little bit of data on one of these compounds from Delos. This is glucose tolerance after, I think, 3 weeks of dosing. I'm sorry, I took this from one of my China talks, and one of my postdocs decided to translate for me, and frankly, I have no idea what they wrote. So it probably says, like, Kennedy doesn't know what he's talking about or something. I should learn the symbol for Kennedy just to make sure it's not in there. So if you do a 3-week dosing with rapamycin and look at glucose tolerance, you can see that rapamycin causes glucose intolerance at this level, 8 mg per kg every other day. At a higher dose, this is chosen to get the same level of TORC1 inhibition. You don't see glucose intolerance with our compound. You can look in disease models. So this is a model of tauopathy that we did through a CRO, and even after only 1 month, you get reduced total tau levels in this model. So there's plenty of diseases where you see efficacy, at least in preclinical models, so we're trying to move these compounds forward. A lot of what my lab is doing right now is trying to get downstream of TOR, though, and ask the question, why is reduced TOR signaling beneficial for aging? And so we've been working to modify factors downstream of TOR, mostly genetically or through small molecules. So we're collaborating with Claudio Hetz in Chile and to look at autophagy, and David Yeast to look at ribosome biogenesis. Very interesting work there. Some of it just published, but it's more developmental, at least what we're studying right here now, and we haven't got aging data yet. And then we're looking at the 2 most studied substrates of TORC1, 4-EBP and S6 kinase. Now, TOR is an activator of S6 kinase, and so if you want to phenocopy the effects of rapamycin, you have to knock out S6 kinase. And so Dominic Withers did this, and we did it independently, and they showed first that the whole body knockout of S6 kinase confers long lifespan in females. but the effect is relatively small compared to rapamycin. And so we think this is certainly not the whole story. And there was no significant effect in males. Both of our groups have been knocking S6 kinase out in specific tissues, and we have yet to find a tissue where it looks like there's a benefit for aging or a response to high-fat diet or the kinds of things you look at with S6 kinase. And so we haven't knocked it out in toenails yet, but we're almost there. I don't know. It may be that you need to knock out S6 kinase in multiple tissues to get this phenotype that was observed for aging. Although I have to say we haven't done full aging studies on every tissue. It's just too expensive. I'm going to tell you more about 4-ABP today. And so TORC1 is an inhibitor of 4-ABP. And so if you want to phenocopy the effects of rapamycin, you need to activate 4-ABP. So we created with Al-Espada flock-stop mice where we can use Cree to turn on 4-ABP expression in any tissue or whole body or wherever we want. And I'll tell you a little bit about those. I'm going to go kind of ahistorically here because we stumbled into a finding that we think relates to sex dependencies with respect to aging. Now, I'm kind of new to this field, and to show you how new, I was going around saying, and we even put in a paper that we're studying gender-specific effects on the TOR pathway, and it took about 2 days for somebody to call me and say, gender is how you identify, sex is biology. And so I just want to assure you that I went into the vivarium and I asked every single mouse whether they identify as male or female. But I'm going to try to use the right language from now on. So if you put, this is a great postdoc, Sheen Tsai, who did both of the 4-ABP studies I'm going to tell you about today. She is now just taken an assistant professor position at National University Singapore. And so she's in her 4th day of work, and I'm still paying for all of her mice, and so I don't know how this works, but to fix that. If you put mice on a high-fat diet, here's the males and here's the females, they both get about the same increase in fat per total body weight. And this is apparently true in humans as well. The interesting thing is that the males have much worse diabetic complications. So if you look at glucose intolerance, there's much more glucose intolerance in the males than there is in the females. And this is really not well understood yet. And one of the reasons it's not well understood is that most of the diabetic studies in the field are done only in males. And part of the reason for that is that they get diabetes faster, but it doesn't help you to understand what the difference is between males and females. So we think some of that relates to the TOR pathway, and part of that comes from this observation, that if you look at 4-ABP in males and females, we have about 20 mice of each sex now. So I'm just showing you a little bit of the data. And this is muscle. You see a loss of 4-ABP expression in response to a high-fat diet. You don't see that in females. In contrast, what we find anyway is that there's higher levels of S6 kinase activity in females in response to a diet than there is in males. So there seems to be sex differences downstream of TOR in this pathway. And we're looking at other lines and trying to see how conserved this is, but this is what we saw in the Black 6 model. And it led us to really begin to try to understand what is it that's causing these differences. And the first candidate for us was really inflammation, because it's known that the males have higher inflammation than the females. This is a high-fat diet now at 6 months of age, and you can see more macrophage infiltration into white adipose of the males than the females. We've also looked at 24 months of age on a normal diet, and there's higher levels of inflammation in the males and the females there too, but you can see there's significant inflammation in the females in older ages on a normal diet. But that led us to try to test whether we could mimic this effect in cell culture. So in other words, can we provide inflammatory agents in cell culture and see regulation of 4-ABP? So we can. So if you take cells, and these are mouse, and expose them to LPS, you get high levels of phospho-JNK, high phospho-JNK levels, and you also begin to rapidly lose 4-ABP expression. So we think what we're trying to do now is we can get this by multiple inflammatory signals in cell culture. There's some differences in different cell lines. There may be differences in different from different sexes of mice, which has interesting implications on what the differences might be. It suggests that either it's a chromosomal difference or it's an epigenetic difference that's preserved in cell culture. but we're really into trying to understand this mechanistically right now. If we look at, yeah.
Are the maps obtained from adult mice or from young?
These are embryos, mouse embryo fibroblasts.
And you see sex differences at that level?
It's suggestive of that, so I didn't show it because I'm still not 100% sure, but it looks like there might be. Yeah. So we're starting to think about doing these crazy experiments that are going to cost a fortune, where we get the mice that have the sex-determining locus on an autosome, so we can begin to separate chromosomes from sex and see what goes with what. Everybody assumes that it's sex hormones that's generating these differences, but if you look at stroke models and some other animal models, it's not always the case, that some of the differences are actually the X chromosome versus the Y chromosome. So I'm kind of agnostic to what's going on there right now. If we look at normal aging, we see something relatively similar. We see reduced 4-ABP expression, this time in liver and fat. We start to see reduced 4-ABP in females in fat. I remember I said there was some inflammation there at that age. And then in the S6 kinase, we see much higher levels of induction in the females than the males in a range of different tissues. So this difference seems to hold true in age as well, and we're really interested in whether this explains some of the, or is a clue at least to explain some of the sex differences you see, because almost every intervention you look at works better in males or females than males, and we really don't know why. Now, we have these mice, I mentioned, that we have a flock stop cassette in front of either wild-type 4-ABP or an activated version of 4-ABP that can't be phosphorylated by mTOR. So this, when we turn it on, is constitutively active, and this is just more of the protein, but still mTOR regulated. I'm going to talk about this mouse first. If we make an activated 4-ABP in the whole body, we get a dead mouse during development, and we're still trying to figure out why. If we put on overexpress the wild-type 4-ABP, we get a small mouse, which is intriguing from an aging perspective, and it has interesting properties. So if you look at this mouse on a high-fat diet, the males are resistant to liver steatosis or fatty liver, not the females. And these are just the mice glucose intolerance studies, just on the mice on the high-fat diet. We don't have the control mice here. They're down here. I just wanted to illustrate this point. So here's the males normally. And when you have the males with 4-ABP overexpressed, it looks more like the females. And so when we've got a lot of data to support this. I'm not showing you because it's published now. When we summarize this, we think that what's happening is that inflammation is driving the loss of 4-ABP. It's more apparent in the males. And when you put 4-ABP back, the male mice are resistant to diabetes. But I don't think this is an overexpression phenotype. I think what we're doing is suppressing the reduced expression you see in the male mice on a high-fat diet. And I think that may be true of aging as well. The male mice are protected from at least phenotypes of aging. They have better muscle maintenance, they maintain better metabolism, and they have less inflammation, suggesting there may be feedback to inflammation as well. So we think this is going to protect the male mice. And so this is how we're thinking about things. That there's really a couple of different things going on here, that maybe more than a couple. This is a simplistic model, where you have aging, which is driving an elevation in mTOR signaling. So that you have chronic signals through the mTOR pathway that are too high in various tissues. And you also have inflammation. And of course, aging is related to inflammation. But the inflammation is higher in the males. And so the more important pathway in the males is the loss of 4-ABP. Whereas in the females, it may be this branch that's more dysregulated, so that you have higher levels of S6K1 activity. And that may start to explain some of these effects, because rapamycin works in both sexes, but of course rapamycin influences both of these pathways. S6 kinase knockout works better in females because it may be better to turn down this aberrant signaling in females. 4-ABP, we don't have survival data yet, but at least for properties of aging, works better in males. And that may be because you need to work on this part of the pathway more. And when you look at some of these other agents that seem to only work in males, a lot of them are anti-inflammatory. And so we think that that may be affecting the inflammation as well. We're beginning to look at that with some of these agents. So that's our simplistic model for what's going on, but at least it gives us something to start to test. Now, briefly, if we put activated 4-ABP in various tissues, We would have guessed when we started this experiment that putting it in adipose would be where you get benefits. But that's not the case. We get an obese mouse when we activate 4-ABP, and we're not sure why that is. When we do it in skeletal muscle, though, we get a small mouse. Not just muscle small, but the mouse is small. And that mouse has interesting properties as well. We published this also recently. It's work from Sheen as well. And I'll just summarize the important point because I want to get on to some other topic just before I close. So we activate 4-ABP in skeletal muscle, and we protect the muscle from high-fat diet and from aging. We think this is partly through an enhancement of translation of PGC1 alpha, which is enhancing respiratory capacity of the muscle, switching fiber type, and helping maintain muscle function. But the thing that was surprising to us is we saw that changing 4-ABP in muscle also protected the liver from high-fat diet and aging, and protected brown fat. So that led us to believe that there's something that the muscle is producing, a myokine, that is responsible for non-cell autonomously protecting other tissues in the body. And so we screened all the different myokines that were out there that we could find, and we only found one that was a big hit, and that was FGF21. So there's much higher levels of RNA for FGF21 in the muscle, higher levels of protein in the muscle, and higher levels in the serum as well of FGF21. And that's interesting because if you remember this data from the Mangelsdorf lab, when you have transgenic FGF21 mice, they're very long-lived. This was not a study done several years ago, and it hasn't been focused on very much, but there's a big impact on lifespan in these mice. And you can also see induction of FGF21 in a whole range of different interventions that are associated with long lifespan. So we're working on the idea now that maybe this is a downstream common factor for a lot of different interventions that affect aging. So we're just in the process now of getting our first 4-ABP FGF21 knockout mice to see if that blocks some of the benefits of 4-ABP, but it's an interesting question about whether it's required for some of these other assays as well. Yeah?
You just showed that knockout 4-ABP1 has a beneficial effect.
Yeah.
How about knockout or tissue-specific knockout?
I believe those are whole body knockouts of the 4-ABP1. That's work from other labs, but that is a bit of a puzzle. It's you can either, in some cases, you get benefits of knocking out 4-ABP1. In our case, we see benefits of activating 4-ABP1, and I don't have a good answer for that. It almost seems like that the TOR pathway is exactly the wrong point for aging, and if you modulate it, you might see benefits either way. But we've been more interested in things that mimic turning down TOR signaling, so we've been activating 4-ABP. Actually, what we're doing right now is we're taking TSC knockout, muscle-specific TSC knockout mice, which we think are an interesting model for Progeria because they have a rapid muscle aging and they have a kyphotic phenotype as well. And we're doing our 4-ABP modification in those mice as well as S6 kinase to see what suppresses the effects of overregulation of TOR in that pathway. So, but 4-ABP somewhat suppresses that phenotype, but it's not complete, and we don't have the data on S6 kinase yet. So, but it's complicated. I agree with you. Yeah.
So, the physiological function of 4-ABP1 is to suppress Progeria.
Yeah.
Have you studied Progeria?
We haven't looked closely at EX40, but what we are doing is like ribosome profiling now to try to see which messages are altered translationally by modulating 4-ABP1. And we think that it's consistent with some of the literature that there might be differential regulation of RNAs that have complex 5' UTRs compared to simple 5' UTRs. And so PGC1 alpha, for instance, we can show enhanced translation when we alter 4-ABP1, and we're looking at looking now in an unbiased way for what are the other factors that are turned up and down. But we haven't looked directly at EI4E. We've done some binding assays with 4-ABP and 4E, but we haven't looked in a whole lot of detail. Yeah.
Is there any changes in cellular senescence in the 4-ABP activation?
We haven't looked at that yet. It's one of those things. It's a really good question, and I've got other people in the lab doing senescence and other assays, and I should get them to talk to each other. It's a good idea. So our working model of TOR is kind of this, that it's not the basal level of TOR in a young animal that's really that related to aging. We think that part of what's driving aging is there are events that are driving the aging process, damage, whatever you like, proteostasis, proteasome change, proteostasis changes, telomere shortening, pick your favorite, oxidative stress, pick your favorite models. We think that a common response to dysfunction in cells is upregulation of TOR. And so we think there's an aberrant regulation of TOR signaling with aging. We've seen this in muscle and heart. It's also been reported in liver. I heard just a discussion earlier about kidney. I think that this may be a common feature of aging. And so we think that the rapamycin may be beneficial because it's suppressing this elevated level of TOR you get with aging. So remember, you have to keep TOR down under conditions where you don't want growth and proliferation, and up when you do. And so it doesn't look like in our hands the induced state is changing that much with aging, but it's the basal state that's creeping up, and so that you're losing that differential regulation in various tissues. And so we're looking for conditions where we can have elevated TOR and look at what the pathology associated with that is. Yeah.
Just going back to the 4ABP one.
Yeah.
How many more times does the male express mTOR than, with the 4ABP1, how many more times than the females do the males express 4ABP in the model?
So the expression levels are pretty similar before you treat them with a high-fat diet. And then we see, maybe an 80% loss of levels of 4-ABP in the males, and no consistent effect in the females.
So your transgenic model,
the transgenic model is much higher. And so I can't rule out the idea that it's an overexpression phenotype. It's just that all of our data is consistent with the idea that the bigger effect is through restoring what's getting lost. But there's certainly higher levels of 4-ABP in our transgenic males. It's higher. Yeah.
So the other question is, do they get fat on high-fat diet, or
They get roughly the same amount of fat to body weight. Yeah.
their glucose metabolism?
Yeah. It's more like the females. I should also point out, if we have a second, we have an hour and a half, right? Is that true? Okay. This is in mice. And one of the things about this difference in diabetic complications is that that's typically true of human females before menopause. After menopause, there's higher levels of inflammation and higher diabetic complications. And so it may be that if this idea is right, it may be that these interventions that affect mostly males in mice, that may be very different because the mice don't go through sort of a classic menopause, and the females may be protected through a much longer period of the mouse lifespan. So it could be that these compounds that I'm speculating are anti-inflammatory may benefit both females and males in human populations. Forgot to make that point. So the other thing to say about this slide is that it's amazing how many disease models have elevated TOR signaling. It seems like everywhere you look, Alzheimer's disease, metabolic diseases, other neurologic diseases, autoimmune diseases. I've started looking for this, lung diseases. I've started looking for this now, and you can find a report of elevated TOR signaling almost everywhere you look. And so I think this is not necessarily only a feature of aging. I think it may be a feature of disease. And so we've been working on disease models where there's elevated TOR signaling as well to try to understand how does aberrant TOR signaling drive pathology? Is it a common mechanism? Is it a different in every case? What's going on? So that's what I'm going to come to. Yeah.
So you could say the same thing about inflammation.
Yeah.
Do you have a way of distinguishing between those two factors?
Well, I think they're intimately linked. And so rapamycin is anti-inflammatory. We did one study where we could show with Simon Melov that giving older mice rapamycin could protect against cardiac hypertrophy. And we did a panel of inflammatory cytokines, and rapamycin turns down a whole range of those. So I don't know how separable those two things really are. So we've been looking at laminopathies. I started working on lamins before the connection to aging, and so our lab's been working on this for a long time. This is kind of the record holder, this gene lamin A, for different kinds of diseases caused by mutations in the same gene. We're up to like 15 different kinds of diseases. These A-type lamins are nuclear intermediate filaments that form the sort of substructure of the nucleus that help maintain organization in the nucleus. These point mutations are all over the board, but they kind of cluster into dystrophic syndromes like muscular dystrophy, cardiomyopathy, and lipodystrophy. And those are likely caused by either recessive or dominant negative mutations in these proteins. Remember, they're filaments, so if you can make a lot of dominant negative mutations that block further filament assembly. And then you have the Progeria syndromes, which are gain of function. And the common mutation that causes progeria is a splice site silent mutation that activates a splicing of 50 amino acids of the C-terminus of lamin A. And the reason that's relevant is that there's a proteolytic cleavage site that gets lost in that splicing event. And that leads to a permanent farnesylation of lamin A on the C-terminus. As opposed to what normally happens in the normal lamin A, which it gets farnesylated. The farnesyl group helps recruit lamin to the nuclear envelope, and then it gets cleaved, so it doesn't have that farnesylation anymore. So sorry, that was a long-winded explanation. I'm only going to show one slide on this. So you might guess I'm going to talk about progeria here, but interestingly, we don't see elevated TOR signaling in this progeria model. But we do see it in the muscular dystrophies and the lipodystrophies, and so I'm going to talk about those. However, there's a lot of speculation that rapamycin might be good for Hutchinson-Gilford progeria. That it's been shown that it corrects some of the nuclear structural defects in cells from fibroblasts from Hutchinson-Gilford patients. And now there's a clinical trial started by Leslie Gordon and the Progeria Foundation and supported by Francis Collins to look at, I think they're using everolimus, in kids with Hutchinson-Gilford progeria syndrome. And so they reached out to us a while back to see if we could see benefits in animal models of HGPS using rapamycin or everolimus, and the answer is no. So this is the enzyme that does the proteolytic cleavage event on lamins, and we don't see any benefit of rapamycin. And maybe more concerningly, here's the actual genetic copy of the human mutation in mice. It's a G609G mutation. It's silent. It has the splicing effect, and it causes expression of progerin in mice. And this is complicated, but this is the part to look at here. Here we're treating with relatively low levels of rapamycin. There's no effect on median survival. Actually, 4 mice died earlier than we've ever seen in this model before, suggesting there might be some toxicity. 1 mouse lived a long time. I don't know what's going on there. So statistically, no difference, but I'm actually a bit concerned about what's going on here. We only looked at 20 mice. And Carlos Lopez-Otten tested at higher concentrations of rapamycin and found it was toxic at higher concentrations. So right now we have no animal data to support any benefit for this drug in Hutchinson-Gilford syndrome. But that doesn't mean that if we tested the right dose, we might see some benefit. I don't know. We're still trying. But I'm a little bit concerned about a clinical trial going forward, and it's already starting. So, that's all I'll say on that. Hope I'm wrong and it works. But if you look in the lamin knockout mice, there's very interesting things going on with the TOR pathway. These mice, first of all, while I'm on complicated things, this was supposed to be a lamin A knockout mouse. And Colin Stewart made this, and it's actually a hypomorph. It expresses a little bit of lamin A. And it's actually good that it's a hypomorph, because if you go back and make the complete knockout, you get a dead mouse. And it's hard to study. So this hypomorphic mouse turns out to be very useful for disease modeling. It dies at 6 to 8 weeks of age. It has the muscular dystrophy phenotypes here, centralized nuclei, disorganized fibers. Sorry, my phone will stop talking to me. And it looks, very much classic syndromes of muscular dystrophy. You see dilated cardiomyopathy in these mice. You see the Charcot-Marie phenotype, and they also get lipodystrophy. And so I'm going to talk about some of these different things. And the main reason is that we saw in the tissues that were affected in this mouse, in the heart, we see elevated TOR signaling. In the skeletal muscle, we see elevated TOR signaling. And in the brown adipose, we see elevated TOR signaling. And so we've now looked a little bit in each of these tissues to say what happens when you get elevated TOR, what's going wrong? Is it one thing or is it different things? And the answer, well, I'll tell you the answer. By the way, these studies are done by Chen Yu Liao, who was a graduate student here with Jim, and is doing great work in my lab. Chen Yu's on the left. So if you treat the mice with rapamycin, you get about a double the extension in survival. Big effect on survival in these mice. You can maintain their body weight longer, and these mice rapidly lose fat content. They have almost no detectable fat content when they die, and rapamycin dramatically increases their fat content. So we got curious as to what was going on. So first thing we did is we put lamin A back in the heart. We had a neat way of doing that, so we just thought, everyone said they die because of heart defects, so we thought, well, if we put lamin A back, that should suppress the early mortality. And this is what we found. We got good expression of lamin A and a very minor increase in their survival. Nowhere near what we saw with rapamycin. Now, and when we looked at the heart, and we published this a while back, what we found is that the elevated TOR signaling is blocking autophagy in the tissue. And rapamycin restores flux, autophagic flux. And we think that's what's going wrong in the heart when you elevate TOR signaling. But the heart didn't seem like it was the whole story, so we started looking at other things. One of the things we started doing is making S6 kinase knockouts and combining that with the lamin knockout mice. It turns out when you completely knock out S6K1, it's not beneficial to these mice. But you can get a survival benefit when you make a heterozygous S6 kinase, which restores TOR signaling to about normal levels through this part of the pathway. And you get a modest effect on survival, nowhere near the effect of rapamycin. And then when we did that in tissues, we found the benefit comes from skeletal muscle. So if we make a heterozygous S6 kinase in skeletal muscle only, we can recapitulate the survival benefit that we get in the heterozygous S6 whole body knockout. So this suggests that you can, if you improve skeletal muscle function, which we can show in these mice, you get a benefit to survival, but it's nowhere as big as rapamycin. And we think there's downstream factors from S6 kinase that are mediating this, whether it's translational or not, we're still trying to establish. So something different seems to be happening in the muscle. And then we got interested in these mice, their energy expenditure, because they're small mice. They have trouble with the ambient temperature in the vivarium. And so we just put them in metabolic cages and looked at their energy expenditure. And so they have very elevated levels of energy expenditure both during the day and the night, and rapamycin suppresses that activity. And that got us looking at adipose tissue. So in the white adipose, we see a dramatic atrophy in these mice, increased lipolysis. We look at the expression of lipolysis enzymes that are elevated. We don't really see a lot of changes in TOR signaling. So we didn't focus as much on that tissue. But in the brown adipose, we see elevated levels of S6 kinase activity for TORC1, elevated levels of AKT phosphorylation for TORC2. This is known to be linked to uncoupling the activity of TOR, and we can see reduced levels of UCP1, so reduced uncoupling, and that's restored by rapamycin. So in the brown fat, what we think is happening is that you're getting much higher levels are ineffective uncoupling. These mice are struggling to maintain body temperature, and they're essentially just burning through fat to try to get enough heat to survive. And so that led to the question of what does thermal neutrality do? And thermal neutrality enhances their survival, but again, nowhere near to the effect of rapamycin, even though the effect on energy expenditure is even bigger than that of rapamycin. So if we put all this together, what we think is happening is that you have three different things going on. There's elevated TOR signaling in three different tissues in this one disease model, but the downstream effects of that may be different. In the heart, you see an inhibition of autophagy associated with elevated TOR. Rapamycin suppresses that. If you put lamin back, you can suppress it as well, and that has some benefit. In the skeletal muscle, we have elevated F6K1 activity. We can suppress that genetically, and we get some improvement in skeletal muscle function, and that enhances survival a little bit. And then in the brown fat, we see high levels of a reduced uncoupling, which is leading to high levels of burning of energy, causing atrophy in the white fat, and you can suppress that by thermoneutrality, and that has a small effect. So what this is suggesting to us is that when you get conditions of elevated TOR signaling, all bets are off as to what the downstream effects that are driving pathology might be. And so for aging, we probably need to identify which tissues this is happening in, and then look specifically at what's going on in each tissue, because it may be different. That's not the response I was looking for, but that seems to be what the data is beginning to tell us.
So in your double males, how does this play out between males and females?
We don't see huge differences in the laminopathy models between males and females. These are really rapid onset diseases, and even though there's muscular dystrophy and dilated cardiomyopathy, we don't see a lot of fibrosis in this model. So we're not sure that this is being driven as much by inflammation as by dysregulation of other pathways. And that leads to the question of how do you get from disruption of a nuclear structural protein to elevated TOR signaling? And we have some clues that it might be through DNA damage, which is higher in these mice, but we haven't put the whole puzzle together yet onto how that happens.
Does the rapamycin treatment of the lamin hypomorph, does it restore nuclear architecture to any extent?
It's hard to look at that in vivo, and we saw very modest effects in cell culture. Now, we didn't look in the Progeria patient fibroblasts like the Collins lab did, so I have, partly on purpose, so I have no idea whether they saw dramatic effects, which may be true, I don't know. But we look more in the hypomorphs or in the disease mutants, and in that context, in cell culture, the effects are relatively marginal in our hands. So. Yeah.
Are the hypomorphs smaller?
Yeah, they're dramatically smaller.
Do you think that would alter the temperature that you need for thermal neutrality so that it's really 30 degrees?
Yeah, it's possible, although the effects on energy expenditure were really dramatic at 30 degrees. So, but that is possible. We don't have this is a problem with most vivariums, is that they're great for us, but they're not so good for the mice. And I think that the only way we could do this experiment is we just kept them in the metabolic cages. We didn't have any other way to modify temperature. So we took one shot at this, and this is what we got. I think that, if I were organizing vivarium now, I'd be looking for ways to change either parts of rooms or whole rooms so that you can do more of experiments at 30 degrees. The effects on aging, there's clues that they're going to be pretty strong effects, and so I think that we probably need to look at that. Either that or we need to put people in the cold, and that's the other option. So if I go back and just summarize where I think the aging field is, I talked about a bunch of approaches already. These are ones that weren't even on my list. Nutritional supplements, there's a lot of talk about this basis and Elysium and NAD precursors. I've been talking to a lot of people in China. I go there a lot about traditional Chinese medicine, and that's there's a lot of promise there. How to get to it is hard to figure out. There's companies being developed like Unity coming out of the Buck with Judy Campisi to look for senolytics or drugs that kill senescent cells or suppress the senescence-associated secretory phenotype. You've got Liz Parrish now doing gene therapy on herself to try to activate her telomerase and suppress her myostatin. And I'm not really an advocate of that, but people are thinking about gene therapy for aging. I saw her recently, and she was very adamant that her muscle mass has improved, and so I don't know, maybe. And there's longevity clinics and medical diets like what Walter Longo's doing. And so there's a lot I don't know which of these are going to work. But there are a lot of candidates out there that I think are intriguing. And now we're thinking about ways to do clinical testing in humans. I'm sure most of you have heard of this HealthSpan trial with metformin that Nir Barzilai and others are leading. the classic approach has always been to try to treat age-related disease with molecules that slow aging, and I've always been hesitant about that because I kind of think of it as this homeostasis thing. You know, you have things that extend homeostasis may not be the same things that work once you get into disequilibrium, but it's that at least there's an easy route to approval there, so that's led a lot of companies to go down that path. Ultimately, though, I think a lot of us have felt that testing these kind of interventions against aging biomarkers is really the way to go, because you don't have to do full lifespan experiments. The problem is that what's a good aging biomarker beyond walking speed? But I think now that's changed. You know, in the last 5 or 10 years, we've got a lot of candidate biomarkers, and I'm optimistic that some of these are going to really tell us about aging. I think the data on the epigenetic clock's really interesting. Several groups are doing metabolomics, P16, inflammatory cytokine panels, telomere link. To me, this is not an easy thing to measure, but there is some promising data. There's other functional markers being developed, and I think ultimately it's going to be systems approaches that put some combination of these things together. But we have candidates now. So when you look at where we are now, we've got potential interventions, we've got potential biomarkers, we're starting to think about regulatory groups listening. We haven't done a whole lot about this yet. Hopefully we can change this, but it's starting to change. I'm really optimistic that we're going to, in the not-too-distant future, start finding things that can work in humans, at least toward the biomarkers. And, you know, I think ultimately we've got to think about it from a holistic perspective. So we can talk about longevity and aging. I didn't even talk about regenerative medicine, although even in my lab now we have several adult stem cell projects going. Obviously we need new treatments, but we clearly also need better wellness and primary care to target this pathway here. You know, we want to get from here to here. We want to do it by extending health span, and I think it's going to be these things further up the list that really have an impact on that. So I'm excited about where we are in the field, and you know, looking forward to trying to figure out what we can do in humans. So the people that did the work in my lab, Chin Yu is doing almost all the work on lamins that I told you about. Yu Mei is also working on the Progeria now in the lab. Xiyin just moved to Singapore and did all the 4-ABP studies. Mark McCormick is a great postdoc who's looking for jobs, and he did all the yeast studies. Stuart has helped out with all the mouse stuff and have a great lab. Lots of collaborators, been able to get funding, luckily, from a lot of agencies. And so thank you for your time. I'm happy to take more questions.